yeah, my name is Alicia Comino. I'm a PhD student in the Center for Magnetic Resonance, Biological and Medical. It's the name in French. It's called CRMM in Marseille, in the south of France. And the talk that I'm going to give today, I decided to call it The Influence of Biological Sex on Cerebral Malaria and Multi-Modality Imaging Approach. Basically, my thesis is about cerebral malaria, which I will go over what it means, but especially the sequelae of this disease, so what happens after, and the influence or the view that we have of sex in this disease. And I will try to walk you over all of these things and make some important points for me in this talk. And for this, I chose some of the results that I have from my thesis to illustrate this point. So we'll start. So first, an overview of what is this disease, what is cerebral malaria. I think we have all heard of malaria. It's it's an infection by the Plasmodium parasite. When the infection is specifically by the Plasmodium falciparum parasite, sometimes also with another type, but specifically the majority of these cases are with this parasite. There is a lethal complication. This is the most lethal complication of this infection, that it's called cerebral malaria. That basically it's a neurological complication. So the parasite arrives and has symptoms in the brain. It's a complication that it's quite relevant to me because it affects especially children under 5 in sub-Saharan Africa, which is a population and a region that, well, are not in the focus of most of the research grants. And it's a complication for which the treatments that are generally used for malaria are normally not really efficient. So normally there's a percentage of efficiency, but most of the times it's just spontaneously or like with palliatives, it gets cured, but still it has a really high mortality. And all of the vaccines, the 2 vaccines that were released for malaria in the last couple of years, only offer 65% of protection, which is not a really high percentage. And essentially, what happens is that this disease is a neurovascular one, which is important because unlike other infectious diseases where the parasite goes into the parenchyma of the brain and leaves abscess and etc., in this case, the parasite never goes into the parenchyma of the brain. So we never find it in the brain tissue. It stays in the microvasculature, and what it does, it sequesters the red blood cells. So basically, it makes the red blood cells stick to the capillaries of the brain. I'm going to see if I can put the pointer. Okay. So basically, you can see here that the parasite makes the red blood cells stick to the capillaries of the brain. And this releases a cascade. Mostly it's an inflammatory cascade, but there's also vascular constriction. There's an activation of the endothelium. There is brain leakage that we can see in microhemorrhages. For example, here in this brain of a child that died of cerebral malaria, you can see there's micro hemorrhages all around the brain. And eventually, importantly, this leads to a rupture of the blood-brain barrier and to a brain edema. That this is an MRI of the brain of a child with cerebral malaria. And as you can see, the difference with a healthy child is really striking because we see that the brain is it's so gonflated, it's so edematic that we don't see all the gyri, and eventually the cerebellum and the whole brain crush the brainstem, and the death is death by a coma, but this happens in 2 to 3 days, so maximum 5 days. So it's a really fast disease also, and it has a 15 to 20% of fatality, so it's a really high percentage. And importantly for us, especially for my work, it is an infectious disease, and one thing that we know about infectious diseases, and well, in general, is that there is a sexual dimorphism in how the host responds to the disease. This is really topic for a big thing. It has many components because, of course, there is a gender component of disease, especially not special, but in in the areas that are endemic for malaria, there is like a really big influence on gender, on like the people that are outside the home, inside the home, the people that access the medical care. This is really important, but also there is a big part that is due to the chromosomes and especially the sex hormones, because estrogens tend to be protective, testosterone is immunosuppressive. So there is an influence that is really seen in infectious diseases, and it leads to different susceptibility to this infection, in this case giving advantage to the female biological sex. However, for cerebral malaria, which is it's a complication of one of the biggest infectious diseases in the world, there is a clear lack of epidemiological information on the sex differences, on prevalence, on severity, and mortality. So it's really hard to find data, this governmental data, official data, on how this disease touches males and females differentially, or if it does. We do have some regional studies, so scientific studies, which are really important, but also the big epidemiological studies made by governments are like really heavy in this case, and we don't find this. But in the regional studies, we can see that there is really differential things going on. We have, for example, studies in Uganda that say that the prevalence of cerebral malaria is much higher in male children, in this case, on this study. But also we have studies in India that say that the mortality on adults over 40 years is much higher in females. So we can see that there is some sex-dependent risk factors that are associated with cerebral malaria, but we do not yet know to which side this goes, and especially we don't know why. And also important for my study, the information and the data I'm going to present is not only that there is some sex differences that we're overlooking, but also cerebral malaria, we said it's a neurological complication, and really importantly, it has neurological sequelae. And also, again, really undiagnosed, really understudied. So we know that they exist for a long time, but it's really hard that these patients are really thoroughly followed. There's a whole battery of sequelae that are very unspecific because they're behavioral sequelae, but also cognitive, motor, sensitive, epilepsy. So we have a lot of sequelae that are linked to this disease that are, well, underfunded, understudied, and well, not even have to mention if there is information on how the sequelae touches its sex differentially. So this was the overall introduction of my project, of a bunch of information that I'm going to present today. So for the objectives, basically, we're going to talk about, okay, what are the sex differences first during the disease. So at the peak of the disease, this is the first thing that we want to know, that we need to know what is happening when the parasite is still in the body. Then we want to know what is happening when the parasite has left the body. So what are the neural bases of the sequelae that we know that they exist, that have been described, even if they're not currently followed, but we know that they exist. And then, are any of the sequelae sex-dependent? And most important, and this is a point that I really want to go through in my talk, because the study of sex differences, if anyone here present works in sex difference, is sometimes too focused on sex differences. So we just want to see the difference, but we don't look at the similarities. So for me, but also for my team and the work that we do, it's important that we focus on, okay, what are the sex-dependent changes that we see, but also what are the sex-independent changes that we see? What are the things that are attached in both sexes, and maybe we can really focus on these ones to study like the sequelae, the potential treatments, etc. So also to and to really think about what these sex-dependent changes mean. So, well, this is a complex diagram of what I do, but I wanted to put it so you would see everything we work on. So to study this, we work in mice. We have a mice model of this disease that is really has been really proven to be a really good model of the disease, especially of the disease in children, of pediatric disease, because it's the first time that they're exposed to the parasite that is the worst, and in a case of our mice, it's the same. It's the first time that they're exposed to this parasite. So we inject these mice with the Plasmodium parasite. And then we follow them first during 7 days, which is when they develop the cerebral syndrome. So 5 to 7 days, still a really fast disease also in this model. And in these days, the animals that have been injected are divided in 2 groups. One group is imaged to see what is happening during the disease, at the very peak of the disease. And then they're euthanized. The other group is treated with the standard treatment for malaria, which is the chloroquine. And then we're going to have 2 control groups. One is going to be a control for the treatment, so it's just going to be injected with chloroquine, and the other one is going to be a saline control. And then these 3 groups that we have injected the treatment, we keep them for 6 months, and we image them once a month with MRI. So the technique that we use to study these sequelae, these sex differences, is going to be multimodal MRI. So we're going to use a bunch of techniques. We have in the lab, we have 2 magnets. So we have one magnet that is vertical, that is 11.75 Tesla, and one magnet that is horizontal that is 7 Tesla. For comparison, in humans, in hospitals, the magnets go from 1.5 to 3 Tesla. And in research, we have up to 7 Tesla, but in mice, when we have them vertical, we can have a lot of Tesla because they're really small magnets. And yes, so we keep these mice 6 months, and then we do once a month imaging. So we can see the progression of the disease. We can see the short-term sequelae, but also the long-term one. And also we do some behavioral tests because we also want to see what's happening to them. What are the sequelae that we have, not only the sequelae in the brain, so the neural bases. So what is this multimodality imaging approach? Well, I'm going to present a bunch of techniques. I'm not going to go specifically into them. If anyone has questions after, we can discuss. But I'm going to give an overall view of the techniques that we use during the peak of the disease. So when the mice are sick, and they're really sick, the only thing we really can do is macrostructural imaging, so anatomical imaging. We can do T2 images, basically images on the brain, and then we treat these images because the mice are very weak, because it's a really, really severe disease. And so we cannot keep them in the magnet for a long time because they're anesthetized with isoflurane. So we really need to be very careful, and they're really, really weak. And yes, so we do only microstructural. So we do volumes. We see the brain, and we have developed an automatic segmentation technique that is based on NNUNet. NNUNet. So it's deep learning. It's a deep learning method. And basically, what we do is we trained it with a lot of our images that we manually segmented, and now it's able to segment the brains in 27 regions automatically. So this gives us, this is a big advantage to us because we can see what is happening in the brain in general, but also we can go to each structure specifically. And then for the mice that we follow for 6 months, we also do this imaging because we also want to see what's happening after. And then we do 2 more things. We have looked at the macrostructure of the brain, so what is happening with the brain regions that we see with our eyes. But we also want to look at what's happening at the microstructure. So what's happening in the gray matter, what's happening in the white matter. And for this, we do DTI. This is diffusion weighted imaging. It's a MRI technique that measures how the water moves through the tissue. We're able to measure the directionality of the water in the tissue, and also if it moves more or less freely and in which direction it does it. So this gives us depending, we have a bunch of measures, and then this gives us the information of, okay, the water diffusion is augmented or is reduced in this direction, so this could mean this could mean that. We'll talk about it later. And then we do functional MRI. So we study the functional connectome. So the connection between the regions, the activation between the regions. And in this case, we do resting state, because fMRI, you can do it with a task. In this case, we don't have a task because we're not interested in any task in particular. We see the spontaneous activation of the different brain regions along the time. So it's a timely correlation. And now I'm going to present some of the results that we had and what this could mean, because I'm less interested in showing you my results and more interested in showing you what we can know when we apply multimodaling technique, because this is a thing that it's really it's done in clinic and it's done in humans, but since you have to have the patient awake on the magnet, it's hard to have a lot of techniques, so you normally do a protocol, and if you put more than one or two techniques in the protocol, it really it becomes really tedious for the patient, and it's really hard to get this information and this acceptance to do this study. So we take advantage that we have a mice model, that we have mice that we can follow the same mice from where they were sick to 6 months after, which is around half the life of a mice. And we can study the progression of all of these parameters. So first, for the peak of the disease, for the cerebral syndrome, we looked at, I said that we only did volumetric data. We looked at 3 biomarkers. They're the most important biomarkers of this disease at the peak, which is first, there is brain edema. So basically, this is the brain of healthy mice, and this is the brain of a sick mouse. So you can see the volume almost duplicates. It's a really visual change. And we don't only see that this volume augments, but we also see lesions. So the kind of lesions that we see are these black points that I point here in blue, and that you can see also here. These are hemorrhages, because it appears as hypointensities in the T2-weighted images. And we also see hyperintensities. So all of these really bright areas, these are lesions in the parenchyma, generally linked to inflammation. So these hyperintensities are another of the lesions that we can observe. And when we see all of these biomarkers together, we have some information. For example, we know it's been described that in this disease there is brain swelling. So I've shown you in the MRI of the child, there's this increase in brain volume that goes, again, there's increase in intracranial pressure. It's really big. And we see in our mice that just this happens. So, okay, our model is valid. So we have in females and in males, we have this big, this increase in brain volume. Between control and peak, there is 3 to 5 days. So it's not due to age. It's really a swelling due to the disease. But we also see, when we look at the 27 regions of the brain that we analyzed, we see that in most of them, the changes that we see are not dependent on sex. So we see that there are changes that are the same in males and in females. For example, a great example is the cortex. The cortex is responsible for 50% of the brain swelling. So basically, the cortex expands, and what's happening is some structures expand with it, but some structures are crushed by it. So we have the big structure, the cortex, the pons, that expand and crush the smaller structures or the structure that are directly under them. But in a lot of the structures, we did find changes that were not dependent on the sex. We did find others that, yes, these changes were more sex-related. For example, we found that the colliculi are crushed in the case of the superior, but edematic in the case of the inferior one. And this is only happens in females, in males. And then we see, for example, for the anterior commissure, we see that in females, it's expanded, it's increased. In males, it's crushed. So this tells us that there are changes that are sex dependent. Okay, we have seen that there are changes that are not, changes that, yes, they are. And if we look at other biomarkers, for example, in the hemorrhages of the brain, we see that if we divide the location of the hemorrhages that are seen, like I see here, as this blue arrows, you can see when, for example, here there's a huge, very clear hemorrhage in the brain. We see that the males have way more hemorrhages in the anterior part of the brain, so in the olfactory bulbs. So we can see here a lot of red, really significantly bigger count of hemorrhages. But some of the females have a bigger count when we go to the posterior part of the brain. So, okay, this is a sex difference. So when we have these kind of results, we have sex-independent changes, sex-dependent changes, and sex differences, what we call, I call it sex-dependent because sex differences, I don't like the term personally, but well. So these sex-dependent changes, we have to think, why are they there? Because me, immediately when I see, it's like, okay, males are more affected, but then we have females that have way more hemorrhages in another part of the brain that males basically do not have in this part of the brain. And then we have regions that are more affected in females, more affected in males. It's a whole puzzle. So what do we see when we look at these results at the peak of the disease? Well, first, I didn't mention, but when we look at when we follow up the animals clinically, we see that some of the clinical signs that, for example, are weight loss, start earlier in the males. So, okay, so they start getting sick before. But then the survival was the same for both animals. The amount of parasitemia that they have in blood was the same for both animals. So what could be happening? Well, to know what could be happening, we look at the biomarkers. And for example, we have a really, for me, a really key biomarker, which is the spatial difference in the lesion distribution. As I say, the males have more hemorrhages in the anterior part of the brain. The females tend to have more in the posterior part. And here we look at the disease. We look at the studies that other people are doing, other imaging studies, and we see that this disease has a progression that is rostrocaudal. So basically, it's a disease that the parasite enters the brain vasculature through the olfactory bulbs. This is the same with other infectious diseases. For example, it happened with COVID. COVID has the same progression, that the pathogen starts its way through the olfactory bulbs because the blood-brain barrier is thinner in there, so it's easier to access. And then it moves to the caudal part of the brain. So it moves to the back. So the last places that it touches are the brainstem, the cerebellum. And this we see also because in the end, these patients of cerebral malaria, what they die of is of crushing of the brainstem, where are all the regions, all the vital functions, like the nuclei that control vital functions. So these are crushed, so there's a coma. So we see that actually what is happening is that the males have more lesions at the start of the disease and females at a part of the disease that is considered the end. So in a way, it's not that the females have more lesions and they're more affected by the disease, but what we can interpret if we know this information is that actually what's happening is that females are surviving a more advanced disease. So they're reaching a later stage of the disease. So males are dying, but they're dying, or they're, because when we image we have a series of parameters where we know that the mice is really at the peak of the disease, is really bad. and then we image them, and then we euthanize it because we don't want the mice to die. We want to get the information, and then we want to humanly sacrifice it because it's really severe disease. So when the mice reach this point that we know they're at the most severe stage, actually the disease has not advanced that much. So the disease has stayed in the initial stages. So we see a lot of mice that have a really hard initial state of the disease. But when the females show the signs of severity, the disease has already progressed all the way to the back. So the lesions have already spread all the way to the back. So here what we are seeing is a difference in severity, but also a difference in resilience to the disease. And then also what we observed with the volumetric data, with the alterations in volume, is that first, there is a regionalization of the edema, so not all brain regions are touched the same. And yes, there is sex-specific alterations, but also most of the regions, we have common alterations. So this was a sex study, but we wanted to focus also on the similarities, because then when we're going to look at the sequelae, we also want to know what are the areas that are touched in both sexes and where the areas that are going to be more affected, like the cortex, the brainstem, etc. So this is what we know. This is the first part of the study at the peak of the disease. Now, as I said, some of the mice that are sick with malaria, we treat and we rescue them. And they're fully rescued, which means that it's a treatment that lasts 10 days. And at the end of these 10 days, there is no more parasite in the blood. So we don't see this parasite. These mice, if they were patients in any hospital, they will be released, they will be considered cured. So this is a thing that happens a lot in infectious diseases. Now less, but it used to happen. It's like when there is no more pathogen, there is no more disease. And actually, a lot of the infectious diseases that touch vital organs, for example, the brain, it's not the case, because you have sequelae, and we have seen it really recently with the long COVID. The parasite, the pathogen is not in the brain. The pathogen is not in the body, but the sequelae are still neurological. They're still important. So there's still important that we look at them, that we study, and that we see if they can ameliorate, if there's something that can be done at the peak or to improve the life of these patients. So for these mice that have survived now this disease, we can look at the clinical biomarkers. This I just say to put in context, like I said before, at the peak of the disease, the male get earlier signs of the disease. In this case, the males recover more slowly. So another biomarker, another clinical marker is the temperature, because they suffer from hypothermia during this disease. The males stay longer in hypothermia, which is not good for the brain. And also the survivor is much lower in the males. So we started with similar cohorts. We end with much less males because they die more. So we look at this data and we say, okay, males get more sick. So now I'm going to show you a way. I'm going to show you examples of some different brain regions, 3 brain regions that I selected. So you see how we can study what is happening in these brain regions using multi-imaging. We can also see what are the sex difference, because we have sex data from all the sex. We analyze together, we analyze separately. But this study was also about seeing, okay, when we have different techniques, what information do these techniques give us? For example, one of the regions that really interested me was the olfactory bulbs, because it's the first region that is touched by the parasite, and also at the peak of the disease is the region where we saw a huge amount of hemorrhages. So it's atrophied, it's hemorrhage, it's basically it's basically a sponge. So at this point, and we can see that these regions in this area, which is this area that you can see here, they do not disappear when the parasite is cleared from the body. So this is a mice before the disease, and this is a mice 1 month, 3 months, and 6 months after the disease. So we see big hemorrhages, we see atrophy. So it really is striking. It's really a sequelae, and it's really a modification in the brain that stays 6 months, which is half the life. So it's an impact in the brain that never recovers. The brain never goes back to how it used to be before a 5-day infection. So also, what do the other techniques that we use, what can they tell us about the olfactory bulbs? For example, we see with the automatic segmentation, we see that both males and females, of course, there is atrophy. It's a total collapse of this region. So there is a really a lower volume 1 month after the disease in both males and females. But in males, the volume starts recovering and goes back to similar volumes to the controls 2 months after the disease. But in the females, the damage is big. The damage is that big that really the volume takes up to 4 months to go back to how it used to be. So the volume recovers a bit, but it really takes much longer. So it's a really longer sequelae. And then if we see another technique, for example, here I show you some results of diffusion-weighted techniques. So here it's the ADC of the olfactory bulbs of females and ADC of the olfactory bulbs of male. ADC is the diffusion coefficient, so it's how much the water diffuses in the tissue. When the ADC, when the diffusion increases, it means that the water has no restriction. So it generally means cell death or any other cause that makes the water, the tissue, to allow the water to pass more. So in this case, we can see that in olfactory bulbs of females, at the first month of this disease, the ADC is really increased, which means that the tissue is very damaged. So we see that the damage at the level of volume, it's bigger in females because they take longer to recover. We also see damage at the level of diffusion that we don't see in males. So we conclude, females have more damage in the olfactory bulbs, but now we know what is happening at the peak. So we really think about it when we have a sex difference. It's really important that we think about why does this sex difference mean? Because if we only stay at females have more damage in the olfactory bulbs, we stay at females are more affected at this level. But then what does this really mean? Because does it mean that there is a sex difference in vulnerability? Do we go look for sex receptors, for sex hormone differences in this part? Or do we think, okay, is this really a difference in vulnerability or is it a difference in resilience? Because for me, this case of the olfactory bulbs, it's really clearly a difference in resilience. Because it means, for me, that the females are getting a more severe disease. And even if they're getting a more severe disease, because this is the start of the disease, we're curing them and they're surviving more than the males. So basically, for me, what this means is they have more damage in a region which was the start of the disease, and even with this damage, they're able to survive. But in males, the only males that survived are males that have the damage is not that bad. So this is just for me, because this is the way I like to think about sex differences, is this. That it doesn't necessarily mean that one of the sexes is worse and one of the sexes. We have to look at the bigger picture. We have to look at what's happening in all of these stages of the disease in all of the different techniques. Another example, for example, we have the hippocampus. In the hippocampus, I found that there is at the end of the disease, not at the beginning, not right after the disease is cured, but 6 months after, suddenly in the females, in all the animals in general, so it has something to do with age, but more in the females, the drop in axial diffusivity is huge. Axial diffusivity is the diffusion of the water, among the axons. So among the axons of the brain. So basically, a drop in axial diffusivity, it means axonal damage. So we are seeing in the females this long-term damage of the axons in the hippocampus that we see only if we follow these mice. So if we only study these mice 1 month after the disease, we don't see this. The hippocampus is not affected. But if we follow them until the end, and this is really a reflect of what happened in humans, because in humans there is some sequelae that appear years after. So there were some studies that were done in children that showed that the children at release have some sequelae, and then the same children years after, 1 year, 2 years after, they have other sequelae that can only be explained by the disease, because they all have them, but that they were not there when they were released from the hospital. So now what we can do, we can combine this result with other results. For example, in this case, I show you a combination with a behavioral test, which is also important. In here, there's a novel object recognition test. Basically, we see if the mice remembers that the object that we put is the one that he has been shown before, or if the animal recognized that the object that was put is new. And in here we see that the females, at the same point where they have this drop, this axonal damage, they're unable to recognize that this object is a new one. So basically they don't go check it. They think it's an object that they have seen before. They don't go check this new object, and this is an indication of damage in the hippocampus. This is an indication that the memory, the short-term memory is not working well. So we have these results in the hippocampus, and the last structure that I'm showing you is the thalamus, because as I said before, the study of sex differences is focused on sex differences, but it's not focused on sex similarities. And yes, it's important that we study what is going on in males, what is going on in females, why is it going on, but also what is going on together. Because I have all my data separated, but I never, like, I rarely think of putting it together, because of course, when you have a different tendency, you don't put it together, you study it separately. But then what happens when you put the data together? For example, when we put the data together, we had a really clear result in the cerebellum of all of the mice with cerebral malaria. We have this big drop of fractional anisotropy. Fractional anisotropy, it's another measure of diffusion weighted images that indicates the organization of the tissue. So the organization of the tissue, it's the organization of the axons, the organization of the white matter, but it's also the organization of the gray matter. And when there is a drop in fractional anisotropy, since that it says that the tissue is more disorganized. So the directionality that the water moves in the tissue becomes a bit anarchic. So we see that at 1 month after the disease in the cerebellum, these all mice have this big drop. And okay, if we combine this technique with another of our technique, in this case, we combine it with resting state fMRI that we haven't talked about before until now. And in resting state fMRI, we see that in the both control groups, there is activation in cerebellar networks. So in these two cerebellar networks, we have activation in these two groups. We have no activation in cerebellar networks that is significant in the malaria mice. So they're having this disorganization of the tissue, and at this same point, when we look at fMRI, we have a less activation on the tissue, less activation in the same organ. So we have seen we see activation in the controls, and we don't see it in this. So this is an area that for me it's really it's really important as an area of sequelae. Because especially if we look at sequelae in humans, one of the top sequelae is motor. It's ataxia, it's paralysis, and in this, especially in ataxia, cerebral ataxia, it's one of the top sequelae that we see short and long term, especially short term. And here we see that 1 month after, this sequelae that is being described in humans, this sequelae that has been described in humans in the cerebellum, it means more disorganization on the tissue, and it means less functional connectivity. So I have a neural basis for this that it really opens up the possibility to explore in these patients. So what do I want to draw as a conclusion from this? is the conclusion of the second part of the study, but also the conclusion that I want you to take home from this study. When I look at my data, what I see is brain sequelae that are regionalized, which it's obvious because we have different cognitive sequelae, we have different motor sequelae, so obviously each is going to touch a different region. But this is a thing that was not known before, that was not studied, because in humans, most of the studies are general brain studies. So it's important to look at the regionalization. And also that for the data that I presented, apparently females are more affected by the disease, because at the level of the olfactory bulbs, at the level of the peak, at the level of the hippocampus, the group that comes out as most affected is the females. But we have to go deeper on this. Why is a group more affected by the other? Does this mean that females are able to survive a more advanced disease? Because they go longer in the disease, or the disease spreads more in them, so they have more severe lesions, but then they survive more, which means that the males that survive are the ones that are having less lesions. So it's really important to look at these big pictures. And also, maybe it's not this, maybe I'm just being delusional here myself, and really there is sex-dependent differences. For example, in the hippocampus, it is known that there is a higher density of, there's a high density of androgen receptors. So maybe we're looking at an effect of sex hormones that are making one structure to be more effective in one of the sexes than the other. But still, the message that I want to pass by is that we have to be aware that the explanation is not always sex hormones. The explanation is not always let's look at the chromosome, let's look at the hormones. Sometimes the explanation is bigger than that. In this case, maybe the explanation is that females survive more. In the case of the humans, maybe the explanation has to be looked at gender and not only sex. So it's really important to look at the bigger picture. And also, the other point that I want to pass through to finish is that if you study sex differences, please study sex difference, first of all. I think it's really important that we include the variable of sex in all of the studies that we do. I think it's really important that we give this vision in all of the projects that we ask and all of the projects that we do, that it is important to have our data, not only including males and females, but studied separately by sex. So separate your data, study different, see what it differs. If it doesn't differ, put it together and report similarities, because it's really also important that we don't not only focus on what is different, but also what there is in common between the two. But having analyzed before, it separately. So with this, I conclude my talk, and I thank my group at the CRMBM. And I am open to answer any question, and I hope this was clear to all of you. So thank you so much for listening.